So, what is chemical space docking? Before we dive into that, these are the travel plans for today. So I will talk a little bit about the concept of chemical space docking itself. We'll just explain the workflow to you. Also for the tech guys here, I will dive into the technical aspects and the requirements to run a chemical space docking. We will have a live demo of chemical space docking to show you how it really performs on the first-hand side. And by the end of the day, I would like also to provide you with some personal perspectives, good practice, and valuable insights, how to actually get the best out of the software. And then I will hand over to Camilla subsequently. So what is chemical space docking? If we reduce chemical spacing to a one-liner, it would be chemical space docking is the next generation of visual screening, right? So we are working with a structure-based visual screening, but it comes with so much more that outperforms standard or common visual screening approaches in almost every single way. Namely, with chemical space docking, you can actually efficiently extract relevant molecules from ultra large chemical spaces. And if I talk about, or we advise that you talk about ultra large chemical spaces, we're talking about billion to trillion sized, and this is not the upper limit. So if the space is larger, chemical space docking can go even farther without crushing your computational resources. What are the key features? Again, ultra large chemical spaces, 10 to the power of 12 is no problem. If you would like to go higher, no issue here. It scales not linearly, but logarithmically. It runs on your hardware, so you're not reliant on any external third parties to share your data. You can do everything in-house. And ultimately, the requirements are quite slim. So you don't need huge server farms. You don't need Amazon computing, Amazon cloud to get stuff done, which is also very expensive. So if you have the infrastructure already there in-house, you can utilize this to the fullest yourself. To have everybody on the same page, we use chemical spaces as a term to describe ultra-large collections of molecules. This is different compared to compound libraries that are normally used to work with. So a lot of people have heard that. So to make things short, compound libraries as an enumerated list, so you know exactly how many entries are there, you know exactly what those entries are. There's an identifier. Everything is listed explicitly. Whereas on the chemical space perspective, it's a combinatorial setup. You have building blocks and you have reactions, and the results are basically generated on the fly. This allows us to be much faster compared to standard methods. Again, full list of all compounds from this space does not exist. Right? This is something we try to avoid. We don't want to enumerate because you will end up with a pile of data that is just very difficult to handle, petabytes for trillions. This doesn't make too much sense, right? So we need smart approaches, and therefore we generate everything on the fly. This is important for you to know you don't miss out. Everything that is inside the chemical space, every potential compound, every entry can emerge with every method you apply. So therefore, nothing is lost. To make things even more digestible for you, enumerated is an endless list, right? So on the left side, if you think about a burger restaurant, an enumerated option would be having a very long menu. Every single possibility of a burger is listed. You have a cheeseburger with cheese, you have a cheeseburger without cheese, you have baked, you have onion. So every combination of those ingredients is basically listed, right? So these are predefined combinations that have to be set up. But given the long list, it will take you a long time to get through every single option to know what you want, right? On the right-hand side, we have the combinatorial setup. We are only aware of buns, toppings, sauces, and extras, and how to combine them, right? So if you have something in mind that you really would like to have based on your flavor profile of the day, you come up to a combinatorial approach, and the perfect thing, or the next closest idea to what you would like to have is generated on the fly. So by combining a lot of ingredients, we get almost infinite options. And the best part is we can actually come up with a solution incredibly fast. So how does this whole idea translate into structure-based visual screening? We start by selecting a chemical space. right? So this chemical space contains building blocks. As you may be aware of every single compound supplier, things that can be coupled with each other based on their functionalities. In the very first step, we dock those building blocks, but we don't dock the exact building blocks, we dock the syntons. What are syntons? Syntons are translated building blocks that have a linker atom, right? So basically, do I have my power pointer? I do. Basically, we translate a functionality, amine, carboxylic acid, and so on, into a linker here. So this linker contains two things. It contains what functionality is there, and also what chemical reaction rules are hidden behind this one, right? What we do in the first step, we take those syntons and we dock them, right? What is the idea behind this one? The idea is we don't have to dock 10 to the power of 12 molecules. This will take a lot of time. But if we work with the smallest fraction of molecules, this is something we can easily handle, right? And once we dock them, we can actually pick out what fragments are even able to survive, okay? So the idea is good fragments that form good interactions, indicated by those green spheres, they are valid starting points because they are kind of like happy inside the binding site, right? They form good interactions. Bad fragments won't end up in good compounds, right? Because if you have bad interactions indicated by the red sphere, this is something that is just not contributing to the overall binding affinity of the final compound as well. So those kind of things is just useless to enumerate. We don't want to see them any longer, or at least this is a starting fragment. So this is where we actually start to remove them from the calculation. We don't want them. We don't want to continue with them. In other cases, we're also asking ourselves, well, if this is the best positioning for the building block, and this is the linker, right? Then this will all extend into this direction. But there is no space. This is where the surface of the target structure begins, so we'll have clashes. This is also something we don't want to pursue. In reality, this is how it looks like. We have compounds that are very happy inside the binding site, green spheres all over, forming beautiful interactions. And we also see that the linker is pointing towards occupied space within the binding site, right? On the other hand, we have structures that are placed somewhere. So they're just floating around, trying to form interactions, or they extend towards the solvent or just pointing towards the surface. We don't want to have them. This is something we would just not continue working with. After we decide what compounds or what fragments, building blocks, syntons, however you want to name them, whatever is legibly selectable as the starting point, is then taken to the next step. In the next step, we now started checking, okay, this contained a particular functionality, amine, carboxylic acid, you name it. And then we ask ourselves, how can we actually combine those with other building blocks? So we start by extending our compounds in the next step. We grow our starting point in green and in blue and add the next building blocks that are allowed to be coupled with a functionality here. So we do enumerate sub-libraries of the starting points and then subsequently dock them into the binding site. Again, we run the same procedure. We check for interactions, clashes, position of the linker, and again select only the best candidates to follow up with for a final extension step. I have to mention here that it's also possible that already some compounds, final compounds, can emerge in this stage. And by the end of the day, this is what you would end up with, a selection of compounds that are good binders and legit compounds you can purchase. So taking this whole concept and translating it into something that is user-friendly, we have designed a very sophisticated workflow for you to work with. This is still some aspects that will be addressed later on, but the core structure is already there and it's very nice to operate. So you're starting with a GUI, with a graphical user interface to prepare the binding site, adjust your parameters, set constraints, and so on and so forth. Then you initiate the anchoring of syntons. You will be presented with the results. So basically every building block that is inside of the chemical space will be docked into the binding site, and then you can download the results and select the best candidates to follow up with. The best part here is you have a lot of visual intervention points where you can select the best candidates. Basically, the traffic light scheme you are most familiar with from SeeSAR to select compounds. Again, you can apply additional filters to select better compounds, but we will address this at a later stage. So, and this whole process of selecting the best candidates to follow up with is possible in each extension step. So if you have symptoms from this stage and you take them over here, again, you are allowed to set new constraints, new pharmacophore constraints to actually check for better potential interactions inside the binding site to keep on going the way you want to. So obviously the question arises, what is the difference of chemical space docking compared to standard visual screening? Okay. First things first, I have an idea that some of you have already run some visual screenings, and there is this sweet spot of few millions, right? Visual screening of few millions, this is something every group can do. But if you think about going to billions or even trillions, so several orders of magnitude more, things become unhandy. This is not something you will be able to handle within a month, not within even a year. So there needs some form of rethinking if you want to work with chemical spaces, large chemical spaces, in-house, right? Another important aspect is that everything included a chemical space is not listed, right? So if you think about it, every single compound inside a chemical space is not mentioned anywhere because the results are only generated when you start a search. So ultimately, everything you get is something that was not mentioned anywhere before, right? And if people kind of like not follow up with a compound, they don't even know it existed. This information is lost. So a lot of novel, unlisted IP is actually hidden inside chemical spaces. As per definition, the results that you actually retrieve with chemical space docking are synthetically accessible. So we are working with a lot of compound providers to construct those chemical spaces. I think enamine is a name to everybody here. So everything you retrieve from those chemical spaces is synthesizable by you, or even better, can be purchased from our partners because this is their business model. And it's a competitive pricing. So all the results you retrieve are synthetically accessible and you can get your hands on it. Now to the second statement here, or the second section, is basically chemical space docking is efficient, and there it is published. I will come to the publications in a few seconds. Chemical space docking is able to accumulate high-ranking compounds. So this is basically a representation of what happened here. And you see that if you perform brute force docking, which is the standard docking method, you end up with a lot of compounds with very bad docking scores. And this number decreases exceptionally fast towards higher binding affinities or docking scores. So you don't get a lot of good compounds. You do get some, yes, but in a lot of 99% of the cases, you're just wasting your time because this compound will not bind and it's just a bad compound for this binding site. Chemical space docking, on the other hand, is able to accumulate good docking scores significantly, just because this is the way of the concept, right? So ultimately, this is green computing. So you are using your computational resources to investigate better compounds. And this is something I have just taken over from the publication to highlight it with numbers. So in this case, 1 million compounds from a random set, so basically a random molecule collection, 1 million compounds from a random molecule collection, full docking, and 1 million compounds from chemical space docking, also randomly selected at the end, were docked once again. And you can see a distribution. In 98.8% of the cases, you got abysmal scores for random molecules, which makes sense, right? You have random molecules without any guidance. So if you find something, it's a unicorn, right? So this is a very, very rare event to find good compounds. Whereas in the case for chemical space docking, you start with very good starting points, right? And you still get novel IP and novel chemotypes and everything. But all of those compounds that you investigate display extremely better docking scores. So in green is the percentage for random docking, and in blue for chemical space docking, and you can see nicely how it is in normal distribution here. So I've talked about our partners, and we do have a lot of them nowadays. So if you go to our page, you can download all of the spaces. There are basically free of charge because they're part of the software. And right now we collaborate with distributors displayed here. The largest one is Explore, which is still a do-it-yourself space, but they can help you to connect you to CROs to synthesize the compounds, while the rest listed up here can do the synthesis for you, and you can order your compounds from them. What people kind of like forget to include in this whole setup is actually that they can also create their own chemical space. So if you have in-house reactions, if you have in-house compounds, building blocks, you can work with them. You can just kind of like get the building blocks from each distributor here, set up your own chemical space, sprinkle on some of your own reactions and compounds, and you will get a completely novel chemical space that can be several sizes larger because this is an exponential growth here, right? So keep in mind that you can also use your own chemical space for chemical space docking. This was published twice already. Basically, one in collaboration with Genentech, which was actually the first publication, but was published later. And this was a collaboration between us, Crystals First, and Enamine. And you can see the hit rates for chemical space docking are good. I mean, they are absolutely decent, outperforming virtual screenings by some numbers. But again, if it's 30% or 40, let's be honest, it doesn't make a huge difference here. And the highlights of both studies are basically good hit rates, very fast follow-up. So basically from starting the chemical space docking approach up until receiving X-ray crystal structures takes only 9 weeks in our case. Almost 14,000-fold gain in affinity in this study, whereas in this study we discovered several novel chemotypes, I think 7 or 8, and also 2 crystal structures were obtained, where here it was 6. So the approach works, right? We know what we do, and it's published science by now. So also others accepted the fact that it's a nice method. Now a little bit into the technical scenarios here. Chemical space docking runs on SeeSAR, but requires another instance for the calculation, which is our platform HPC. So what happens here? We start our docking setup with SeeSAR. This is the graphical user interface for the modeling, for the binding site definition, to apply pharmacophore constraints, and also do the subsequent visual assessment. So everything where you really interact with compounds, where you interact with the structure, where you look at things, save stuff, it happens here. All right, it happens on your own machine, and this is what we call a client. Yeah, so this is basically your terminal to communicate with results. And oops, sorry. And on the left side, we have another machine. This is a second machine that runs HPC. And this is the instance that is responsible for the calculations, right? So this one does the generation of poses, the docking, the enumeration part. Everything happens here, also the space upload. This is also where you can orchestrate your team, set up accounts for users, managing an admin. You can also upload your own libraries for remote docking. So this is where the stuff happens that you don't want to see live, right? And the way it goes is you go to SeeSAR, you start your chemical space docking mode. Then after doing everything, you just say, hey, I would like to start my calculations. You send your calculations to the server. The server does its magic. And then once everything has finished, provides you with results. So this is kind of like the environment that we came up with to make things sophisticated and seamless. A little bit about the technical requirements. The good news, let's start with the right side here. SeeSAR is your communication point, right? You don't need a super machine to do stuff. You basically just need to look at things, turn them a little bit around. So the requirements here are pretty slim. 8 GB of RAM is perfect. Anything else is just better. So, and you need a local graphic card, obviously. The good thing is you can start the whole workflow from every single operating system. Linux, Windows, Mac OS, so the most prominent ones are supported. So for you as an end user, this is the most important part. The left part is a little bit more interesting for the IT guys, or if you have to set it up yourself. So basically, since this is the server, so this is where the calculation happens, you need a little bit more of raw power to do stuff. The minimum amount of RAM for calculating stuff is 16 gigabyte CPU. The more you have, the faster you get results, obviously. Since you get a lot of poses, you need at least 10 gigabytes per active CSD workflow and minimum of 50 gigabytes. At some point, you have to work with some millions, but again, this is manageable. And for cores, 16 cores will lead you to around 10 days of calculations. To summarize the lower part, you have to install HPC on either Linux or Windows. So these are the two systems that are supported. Mac OS is not officially currently supported. I've heard that it is somewhat realizable, yet the people we have talked to so far are using Windows or Linux as their server system. So yeah. But to give you an idea about how fast things can be with chemical space docking, this is a use case that we have examined in-house, namely with Enumerated real space, 70 billion of compounds. So we are using 11 Windows with 64 gigabytes of RAM each, which concludes to 352 threads here. And if we initiate the chemical space docking, we are docking 1 million compounds here. So this is kind of like those are the building blocks that we have to work with. This is the most time-consuming step in theory. So after docking 1 million compounds, around 280,000 survive. And this whole process takes us, with this setup, approximately 8.4 hours. Subsequently, we select 100 compounds, 100 fragments, and enumerate every single sublibrary for each compound. So basically the grown structures for the subsequent docking. And then we perform template-based docking. So this is nice to know and quite important to know. Once you have selected your compound, anything in the extension steps will be docked and is in a template-based mode, right? This accelerates the whole procedure by a factor of 8 or 10. And for 3.6 million molecules, it takes us only 16 hours approximately to finish. The last step, so basically after selecting 50 fragments from those 3.6 million and then finishing the last extension step, we end up with 149,000 compounds, and it does take us less than an hour to dock them. So with our setup that is not huge, I would say this is comparatively normal for a small or mid-sized company. It takes us only almost 1 day for the calculations only to run the whole thing, which is, looking at this number, a very, 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 very nice number. Obviously, I have to mention that those steps here are affected by how many fragments you select in each step. But again, you can kind of like imagine that it's not exploding, right? In these stages, it scales linearly with your fragments. Not every time, because it also depends on the sublibrary number, sublibrary size, but ultimately manageable. Okay, for you guys, if you would like to set up chemical space docking by yourself, we have a CSD starter package, basically. I just want to mention here that you are aware of it. You can also access it from biosafety.de/csd/help. It concludes a lot of PDFs to help you, basically a good practice PDF, collecting a lot of insights you should consider during the selection steps, how to install HPC, and obviously how to operate within SeeSAR. But don't worry, I will show it you in the next few seconds. And also a ROC1 test space that you can use to experiment if your setup is good or not. So a little bit about the test space. Basically, we want to have something very small that can be processed kind of like on the fly, not a huge chemical space, something in the size of 1,000. And I was very uncreative here. So I just looked at random ROC1 kinase structures and took a look what are prominent hinge binders here and what other structures occupy in the middle and basically towards the VFG motif. And I went through the PDBs and selected some known binders basically, and for each one of those sections, and 5 that are not confirmed, that are a little bit more chemically diverse. So ultimately, you now have 5 good building blocks, 5 unconfirmed building blocks, making 10 building blocks for each set, and 10 times 10 times 10 results in a chemical space in the size of 1000. So something you can do really on the fly within 5 minutes on your computer just to play around with the structure. It doesn't need to be used on a kinase. Completely transparent here. You can use it on any structure. This is what the diversity is here for, just to have flexible compounds to see if the docking setup works for you. It can be run before starting a huge chemical space docking. So this was the general introduction into chemical space docking so far. And now I will dive into the live session. And here we are. I hope you still do see my screen. At least I see my screen. So it starts the same way every project in SeeSAR starts. You load your structure and you define a binding site. Here I'm running already on a predefined setup just to have the visuals in the right place. And once we can also take a look for the structure so we are all get familiar with it. A hinge binder. Nothing surprising here. We have the hinge binding motif here, and then project towards the DFG motif in the back end here. Ultimately, 2 ring systems here, 1 ring system there, and 1 terminal ring system in this section. I would say a very, I wouldn't say uninspiring, but not completely overcrowded compound with a lot of decorations. So let's see how the chemical space docking works. To do so, we hover over the mode selector and change towards the chemical space docking mode. I'm already connected with HPC, so before I am able to do anything, I have to go to systems and connect to my HPC client. We also have uploaded a tutorial on that in the with the release of SeeSAR 14. So I'm already online, so I would phrase it. And the first thing we have to do to start the chemical space docking process is to select a space. Right now, I'm selecting the block 1 test space, and I will start my docking. So now I'm basically started a project on the server. Now it knows, okay, this project, this structure is connected to the block 1 test space, and I am in the anchoring step. So I would like to start my anchoring step. This is where I place my sentence in the binding site. And once I have clicked on it, the docking procedure starts. So the building blocks are now taken from the server and placed inside my binding site. Everything happens now in St. Augustine. Right now I'm in Cologne. I'm doing everything remotely, and I'm waiting for the results to finish, which will take approximately 1 minute. So 3D coordinates are processed. The whole binding site is sampled. Yeah. There's also a possibility for me to define pharmacophore constraints before starting the docking. So if I would like to introduce a hinge binder, a constraint, I can do it here as well before starting the docking. Also adjusting the usual docking parameters, like how many poses would I like to generate, the clash tolerance, and also the chair sampling here of 6-member twin systems. And we are almost there. Another few seconds. And there we go. So I'm getting informed my chemical space docking results are there. And if I want to view them, I just click on here. And now I'm downloading my results. So here we are. What do we see? We see that our fragments have been docked, and we have 136 fragments on the server. So basically, we have docked 30 of our building blocks, because it was only 30 building blocks, as you can see here as well, 30 fragments. And for those 30 fragments, we get 156 poses. What we can do now, we can go through the results and visually check on them. So this is the first thing. Now you can see that what I mean. The linker is positioned here. So this is not the site I actually would like to go into. I know my binding site. I know I want to go there. So therefore, this is not something I would like to go with. Another important factor is all of those molecules that I see now are pre-filtered. So you can see it here. Some filters were applied, namely 3, and I open them up. I will see which filters were selected, namely torsion quality, clashes between molecules and the binding site, and clashes of the molecule with itself. Those are the 3 things we apply here. We don't allow anything where we say this is too much. Okay, so we only get green and orange results in this section. It is also possible to apply your own filters here. So if you say, I would like to have only compounds with a particular molecular weight, you can adjust it here. Then say, I would like to have only compounds less than 300 Dalton if you are into a fragment-based project, and apply the filters. Then everything inside this table will be filtered out also on the server. So you will retrieve only those results that are compliant with your filters, and you need to re-download them again. So the filtering happens on the server side, not on the SeeSAR side. But let's not do that at the moment. So let's look at the structures. So this is something we have seen before, right? So this is something we would like to work with. If you would like to work with this fragment or this symptom, we have to select it here, add molecule as input for the next step. We click on it, and what we notice here is that it will be added to our fragment selection window here. Let's maybe pick a few more. I'll just browse through them one by one. Again, I don't want a linker here. I don't want a linker here. This is a new motif. I like this one. Let's add it to the selection. People are also asking, is there a possibility to actually define a placement for the linker? Yes, it's possible. You can place a pharmacophore constraint and say, I would like to have a linker in this position to kind of like guide your docking procedure. So again, so we can actually also work with this kind of stuff, right? So ultimately, we are not starting with H-binder because we didn't apply any constraints, but we can kind of like think about if you would like to proceed with this one because it points towards where we would like to go into. We would like to occupy this space. So let's pick this one as well. Also a nice hinge binder. Let's see if I can find something else. Just for a 5th one. So this is again, it works the same way as you would do in every chemical space, in every virtual screening. If you don't like a structure, don't pick it. For me, it's kind of like I'm not a huge fan of this amide as a hinge binding motif, although the linker is there. So therefore, I will just skip it. I don't need to look at those results. Also keep in mind that the spheres themselves also help you quite a lot to actually get a feeling for a fragment, right? Fragments should be happy. This is the absolute baseline for fragment-based approaches. Fragments should be happy. With our spheres indications, you can actually see how happy a fragment is. If you have a very small compound, from my perspective, the compound should be all green. Otherwise, I would not select it. If the building block itself is a little bit larger, it's fine if some parts are not completely satisfied. But for small fragments, it's quite important to have perfect green sphere distributions. So you can also see if we have like middle compounds or fragments with building blocks that will be connected from 2 sides, they will be also presented with 2 linkers. So you know, okay, in the next step, it will be either go from this direction or this direction. So basically also those multifunctional linkers are accessible for you. Maybe another hinge binder. Let's see if I can find something. Ah, there you go. Maybe that's it. So now I have selected 5 of them. I have selected 5 of them. We get also results like predicted LLE and LE based on our affinity prediction. And now I could again apply pharmacophore constraints for the next step, or as I will not do now, I will do nothing. And I will start my extension step. So now I'll start the extension step. And what happens in the extension step? I go into each fragment, and I enumerate every single possibility of this fragment inside the chemical space, right? So those libraries can become quite huge. Right now, it will be basically 500 because I've selected 5 different fragments. 5 times 10 times 10. And what happens now? Everything will be docked template-based on my initial structure. The results are already there. So you will see what I mean once I download the results. So since I have docked everything template-based, let's go to the top. Since I have docked everything template-based, all my initial positioning of my fragment will remain the same also in the subsequent compounds. This is quite important to know. So again, I can screen and decide whatever I would like to follow up with. So let's pick this compound, maybe this compound. Let's pick like 5 or 10. I really like this one, just to see how fast everything can be. Let's pick this one. Also a nice additional interaction there. Like this one, everything's happy. But be generous, right? So at this stage, it's completely fine to go full out. I will also tell you the numbers of how many you should pick in the next steps. Let's maybe pick this one. Oh, it's not red. I don't want it. Oh, why not? Let's pick this one. Let's pick this one. And maybe I just get another one. Is there maybe that tool somewhere? Ah, there we go. I just picked this one for the funnies. So again, I have selected the compounds I would like to work with, and I'm already entering the last stage, namely the extension step 2. And there we go. Should also take not much more than 1 minute. So if you have questions, feel free to ask them. So right now we're just enumerating the last libraries for those 10 compounds. So basically we're just working with another 100 compounds. Almost finished, 75%. The question is how to actually work with a linker constraint. It's actually easy. It's basically a pharmacophore constraint. You can set a very large area, say, I would like to have this linker somewhere in this direction. You can make it very large, 5 angstrom or so, and just place it somewhere. And the good part about the docking approach is we generate poses until the constraints are fulfilled. So the idea is not that we generate poses and the pharmacophore constraint then filters out poses that are not compliant with the setup. But if you say, I would like to have 10 poses that have the linker here, then you will get 10 poses that have the linker here. This will take more time because more poses need to be generated, but ultimately you will get 10 poses. Right? So it's not a post filtering, it's a pre-filtering step. The results are there. And the results look nicely. So maybe some of you can recall the initial fragment that we started with. And honestly, I've seen a lot of PDBs, and basically we do cover every single PDB combination that is there with the space, but it's also the setup, to be fair. But it's the proof of concept. And those are the compounds that we have generated. We can also select them subsequently and just add them to our final selection. So you see already how many molecules are in there. We can again filter how much we like to see and what we like to work with. Let's take this one, take this one. And yeah, basically, this is the whole chemical space docking workflow. Again, this is just an example space. You will get obviously better results if you work with the full-fledged stuff, but it's easy, right? It's fun to use and it's quite interactive. So I also promised you some insights. So, good practice and valuable insights. You should be aware that it's also possible to use an initial fragment as a template. This can be a co-crystallized ligand. This can be also a generated docking pose. You just have to click on your ligand and then use it as a template in chemical space docking mode. This will then allow you to perform a template-based docking on the whole setup. And our template-based method is maximum common substructure method of minimum 5 heavy atoms. So since you're providing a starting point for subsequent docking, it increases the speed by a factor of 8 to 10. And what's also quite nice, this MCS feature of the template-based stuff allows potential fuzziness, right? So you really don't have to perfectly match your initial compound, but even with 6 or 5 member twin systems, it's more than enough to actually start the whole build-up of the compounds way faster. So a lot of people do that just to accelerate stuff. I recommend you to try it once or twice to get a feeling for that as well. The next part is the linker atom. There are a lot of special magic happening in the technical scenery behind that you don't see. So pay attention where the linker is, as I've mentioned. Is it a dead end where the linker is placed? Is the linker pointing towards the solvent? You don't want to extend towards the solvent because this is where no interactions will happen. So don't do that. Again, unwanted direction. Sometimes you don't want to particularly go into this direction. So therefore, to really guide your search, apply a linker constraint. You don't lose anything, you can only gain stuff on that. A little bit on the background of the linker. The linker is constructed basically as a rotating ethyl group that is not there. What do I mean by that? By being an phantom ethyl group, it samples the space around it. So this is actually there to avoid clashes within the binding sites. We introduced that to avoid poses where the linker is just pointing towards a very small binding cavity. This samples the space made better, right? So it's the results that you retrieve from that are more reliable. The linker itself is chemically inert. It does not contribute anything to the docking score. And it also does not represent the minimum product. There's a lot of magic happening here, but this is actually a very important part because it helps a lot with subsequent formation of functional groups. So an amine, would you dock an amine in an neutral state or an uncharged state, in an uncharged state or uncharged state? It depends on the chemistry, right? And we actually encode this chemistry. So the linker you will see, the amine, it will be also the same amine that will be present in the product subsequently. Sentence selection, so basically how many things should you select for each step? We recommend 50 to 150 anchors for starting points. And in the extension step, you can go with how many you want. The sky is the limit. Yet you shouldn't go above 300 in the first anchoring step because things become quite large and you can easily reach into few, I don't know, 15 millions or so. Just be aware that 50 to 150 is a very nice spot from the handling of the poses. What people should be aware is actually that the scoring itself favors larger anchors. What do I mean by that? Some building blocks are quite large by themselves, right? Because they can form a lot of interactions. Because they can form a lot of interactions, because they have a lot of heavy atoms, the score will increase subsequently. So I also recommend, once you have go through the list for selecting the best ranking compounds, also do this setup with good LE and lower molecular weight and a particular number of H bonds you would like to see with at least 1 to 2 for smaller fragments, because this way an imidazole, as I've shown you in the live demo, would not pop up in the top ranks, right? But it's still a valid compound, it's still useful, but because the score will be quite low, you should go with this setup to actually look at smaller compounds, which are absolutely valid starting points as well. And again, and each step, when you select stuff, pay attention to chemical diversity to just enrich the number of chemotypes you're working with, just as you would do in visual screening generally. Another thing that people are not completely aware of, the chemical diversity is hidden. So if you have kind of like this 3 compounds and you're asking yourself, which one should I pick? Pick all 3 of them. Because the size of the sublibrary can be very different, they can have completely different protecting groups, chemistry hidden within the linker. So as you can see here, this one contains 43,000 compounds and the other 2 lower numbers. So basically, if you are unsure, be generous, pick more syntons in each step. We have reached the final part from my side before we dive into Camilla's talk. So what does chemical space docking do? Chemical space docking is working with only a fraction of compounds, giving you better results, but still screening the whole chemical space ultimately. Every step allows you to perform active intervention, apply new pharmacophore constraints, filter for particular stuff. You can work with co-complex ligands, which be fragments, small molecules, in both predictions to enrich promising candidates to guide your procedure. And ultimately, the assessment of results is very flexible. So again, you have this SeeSAR convenience of traffic light schemes to select the things you would like to work with. And you also are provided with a lot of parameters to actually make confident decisions.
Okay, good afternoon, everyone. As Alex kindly introduced me, I'm Camilla. I'm a very young PhD student, and I'm working at the Helmholtz Center for Infection Research in Braunschweig, Germany. And today I will show you the application of chemical space docking on antibiotic drug discovery. So targeting ISP, which is an enzyme on the MAP pathway. Let me just, okay. So in an ideal situation, upon when you get an infection, you take an antibiotics, which is able to inhibit cells grow, bacterial grow, and you're cured. The problem arises when the bacterium itself develop mechanisms that are able to inhibit the antibiotic function, leading to antimicrobial resistance, which is a huge health problem and is estimated to cause over 30,000 deaths per year, basically meaning that we need new antibiotics. And one of the approaches we can do is to try to target unexplored proteins within the bacterium. Trying to find an unexplored protein within the bacterium, we can think about isoprenoids, which are a huge chemical diverse class of secondary metabolites with a lot of functions and application, starting from the cell membrane to isoprenoids to steroids hormones production, as well as in industrial applications such as the production of squalane and geraniol, which are very important in the cosmetic industry. Despite the chemical diversity, all the isoprenoids arise from two structures, which are dimethyl pyrophosphate and isopentenyl pyrophosphate. What's interesting here is that there are two different biosynthetic routes for producing them. The first one, as the first discovered one, is the mevalonate pathway, which is used by higher plants, mammals, so humans, and some gram-positive bacteria, whereas gram-negative bacteria, which are more concerning as for antimicrobial resistance, some other gram-positive bacteria and plastidial parasites, such as Plasmodium falciparum, utilize the MAP pathway. What was demonstrated, it was that by inhibiting the enzyme of the MAP pathway, you're able to inhibit also bacterial growth. If we take a look at the metabolites and enzymes of the MAP pathway, we can see that there's just one enzyme that takes just one product and consumes it to produce one takes one substrate to produce just one product, which basically means that if you inhibit one of those compounds, one of those enzymes, you also cannot replace its function by any other genome within the bacterial genome. In my PhD, I focused on ISP, which is the fourth enzyme of the pathway, and it's a kinase that catalyzes the ATP-dependent phosphorylation of CDPME into CDPMEP. It has a polar and relatively solvent exposed binding site divided into ATP and CDP-ME binding pockets that you can see here in the picture, and you can also see more or less how the binding of the two substrates are. As you may have already interfered by now, the aim of the project was to combine fragment and structure-based drug discovery to identify new inhibitors targeting Pseudomonas aeruginosa ISP, and what better application than the chemical space docking. I will not go into the details of the workflow because Alex was already extremely good in presenting that. I will just mention that the calculations were performed by Dr. Raphael Klein from Biosolve IT, and that we performed the anchoring using the enamine real space and targeting the ATP binding site ISP. After that, we performed the combinatorial extension and template docked the virtual products into the active site of the kinase using the fragments binding poses as a template. And after that, validated the compounds in vitro. So to define the pharmacophore for the anchoring part, we started from a structure of Aquifex ISP in complex with ATP that was obtained by docking. And from the knowledge that ATP binds to ISP in an unusual sync conformation, and you can see here in the picture on the right, as the adenine, let's say, pointing towards, so oriented towards the ribose, and the phosphate rider, the anti-conformation that points on the outside. So it has an 80-degree rotation, as we can see here on the adenine, in a less energetically favorable conformation. So we selected 3 positive constraints to direct the poses of our docking, starting from the non-optional one around asparagine 60 and 2 optional on residue 61 and 85. A negative constraint not to have the poses solvent exposed, as Alex was mentioning before, and another optional constraint towards threonine 171 to possibly position a heavy atom there. For the extension part, considering that the data are not published yet, so please don't steal them, I cannot show you the structure and I cannot tell you much. I will just say that we started from 200 fragments, a selection of 200 fragments to perform the combinatorial extension, and after that we selected the best 100 virtual products. What's maybe more interesting for this talk, for this part of the presentation, and what I can tell you about is the in vitro validation. So we take all the virtual products, selected 100 poses, and then farther restricted and purchased 42 diverse chemically diverse molecules from enamine and tested them for Pseudomonas aeruginosa ISP in vitro. We decided to use a luminescence-based assay in which I will show you now the results, and you basically measure the unreacted ATP after a kinase reaction. So low luminescence means that the protein is active, high luminescence means that the protein is inhibited. In principle, any compound leading to an increase in luminescence can be defined as a positive result. But in practice, we selected and we set an arbitrary threshold, or at least 40 to 50% of increase in luminescence to define 14 hits, and leading also to all a set of compounds which do increase the luminescence but do not reach the threshold, and we will call them for now gray zone compounds. After the primary assay, we also wanted to validate the hits, and we took all the positive hits plus one randomly selected compound from this gray zone compound that it will be hereafter called C9. We also tested again on Pseudomonas aeruginosa ISP performing triplicate with a continuous assay, which is the most classical pyruvate kinase lactate dehydrogenase, which is a continuous assay. Therefore, I both measured the absorbance at the linear phase of the reaction, but also at the completeness, just to have a better comparison with the primary assay, which is an endpoint assay. 5 of the hits tests were confirmed as active, including, interestingly, this C9 compound, which was the gray zone compound, which was discarded at the beginning. So last but not least, here is the binding poses of the compounds selected. We can see that they bind to the ATP binding pocket, but interestingly, C9, 12, and 18 seem also to protrude to the CDPME binding pocket, and this could help in providing selectivity towards the target we want. As I was mentioning before, all of the compounds also fulfill the mandatory restraint of binding to asparagine 60, and moreover, 3 of them also had an extra interaction with asparagine 99, that it's another residue involved in orienting the adenine in the ATP binding site of ISP. Again, as I was mentioning before, C9, 12, and 18 also seem to protrude to the CDPME binding pocket of the active site via serine 128 or 238, or interestingly, this C9, let me just use a pointer real quick, also on the catalytically active aspartate 130. This brings me to the end of my talk, in which I will just summarize that with this chemical space docking project, we could see that also the ATP binding site of ISP is probably a promising direct target due to the sync formation of the adenine, and that by applying this CST, that combining fragment and structure-based drug discovery, we were able to identify 5 compounds as active molecules out of the 42 that we tested, with a 12% hit rate, that, considering the early stage of the project, is a promising result, further demonstrating that this chemical space docking is a powerful tool for drug discovery, even on a challenging target. What comes after, it's obviously the characterization of how those compounds bind, KD, IC50, crystallographic studies to confirm the docking poses of the active molecules, and hopefully, probably also to try to do some essays on this gray zone compound and analyze the structures to ultimately perform hit to lead optimization. This brings me to my very end of my talk, in which I would just like to thank my host organization, which is the HZI, my supervisor, Prof. Blankenfeld, as well as all the teams, members of my group, as well as the Biosolve IT, in particular, Dr. Raphael Klein, who performed the calculation of this project, and also you for your attention. Thank you.